Today's first open access paper highlight is titled Dispersal Strategies Shape Persistence and Evolution of Human Gut Bacteria, and it's presented by Falk Hildebrand of Quadram Institute Bioscience at Norwich in the UK.
Good afternoon. It is my pleasure to be discussing with you today dispersal strategies shape persistence and evolution of the human gut bacteria. In this work I want to present here, we were looking at the persistence of gut bacteria and whether this is due to continued survival or recolonizations of the same human host. To analyze this question, we were doing a meta-analysis that contained over 5,000 fecal metagenomes, over 2,000 individuals, hundreds of families, dozens of countries, all of this de novo assembled, metagenomic assembled genomes were then reconstructed and delineated into strains. And you can see in total over 1,200 species that were analyzed. And we find in general that there's three different dispersal strategies. Especially interesting to us were tenacious bacteria. These are highly adapted to host and sadly also susceptible to antibiotic treatments. And I hope we can discuss this later in the meeting room.
The next open access paper highlight is titled Statistical Approaches for Differential Expression Analysis in Metatranscriptomics, presented by Yancong Zhang from the Harvard T.H. Chan School of Public Health in Boston, USA.
Hello everyone, my great honor to present our recent work on differential expression using metatranscriptomics data. MTX data has been highly influenced by technical zero and biological zero. In the community, increasing gene copies could increase the transcript abundance in kin. So RNA level can change both because of the regulation and because the microbe grow or die. Here we developed a 6 model to test the differential expression analysis using community data. We found that if you don't have the paired metagenomics data, normalized within species and adjust the total RNA could be a reasonable way to do the differential expression analysis. If you do have paired metagenomics data, M4 and M6 could behave well. However, using the DNA covariate model could provide you more robust to zero inflations than the ratio model. I'm happy to take questions. Thanks for your attention.
The next open access paper highlight is titled Precise Phylogenetic Analysis of Microbial Isolates and Genomes from Metagenomes using PhyloPhlAn 3, presented by Francesco Asnicar from the University of Trento, Italy.
In the past couple of years, we saw an exponential increase of genomes reconstructed from metagenomes that are made publicly available, and for which basically there are no other information than their genomic sequences. So how can we study and characterize them? With phylogenetic analysis, for instance, we can put into phylogenetic context both isolate genomes and genomes with partial genomic information like MAGs. And for this, we developed PhyloPhlAn. And one of its uses is the assignment of a taxonomic label to both genomes and MAG by exploiting the SGB system, which is the largest collection of genomes and MAGs that are clustered at the species level. With PhyloPhlAn, we can also reconstruct different types of phylogenetic trees, ranging from species-specific to tree of life-size phylogenies, which can provide different level of resolution to our genomes and MAGs we are studying. If you're interested in knowing more about where your genomes and MAGs are phylogenetically placed and how PhyloPhlAn can help you, feel free to join my room to further discuss this. Thank you.
The next open access paper highlight is titled Newly Explored Faecalibacterium Diversity is Connected to Age, Lifestyle, Geography, and Disease, presented by Francesca de Filippis from the University of Naples Federico II in Italy.
Good morning or good afternoon to everybody. In this work, we compared more than 3,000 Faecalibacterium-like genome, either from isolated strains or assembled from human and animal gut metagenome. Even if to date only one species is known within the Faecalibacterium genus, that is Faecalibacterium prausnitzii, we identified at least 22 different Faecalibacterium-like species based on their genomic diversity. Among them, 11 species are widely spread in the human gut across different populations and may also occur in the same subject. Indeed, we highlighted that higher Faecalibacterium diversity is present in non-Western populations compared with Westernized subjects, and a reduction of Faecalibacterium diversity also occur in inflammatory conditions such as obesity, colorectal cancer, and IBD.
And our final open access paper highlight of this session is titled Dental Biofilm Microbiota Dysbiosis is associated with the risk of acute graft-versus-host disease after Allogeneic Hematopoietic stem cell transplantation, presented by Vitor Heidrich from Hospital Sírio-Libanês in Sao Paulo, Brazil.
Hi everyone, thank you for this opportunity. I am Vitor, a PhD student at University of Sao Paulo, Brazil, and I'm happy to talk about this recent paper from our lab published at Frontiers in Immunology, in which we profiled the dental biofilm microbiota of patients undergoing allogeneic hematopoietic stem cell transplant. We showed that there are quite dramatic alterations in the dental microbiota during transplant, including loss of diversity, decrease of commensal, and increase of potentially pathogenic bacteria. In a subset of these patients, we also see a huge increase in Enterococcus faecalis abundance, which is an already documented event for the gut microbiota of transplantation recipients, but not for the teeth as far as we know. And just like for the gut, we see a strong association between dental Enterococcus faecalis and a higher risk of graft-versus-host disease after transplant. So because of the easier access to oral samples, the profiling of the dental biofilm microbiota may be useful in the clinic to assess the risk of stem cell transplant complications. I'm looking forward to talking more detail to some of you in the breakout session. Thank you.